Hello everyone, my name is Nitin Joseph, and in today's lecture we will cover about apoptosis, which is from the paper Advanced Cell Biology. Okay, so whenever we hear apoptosis, it sounds familiar because in cell biology we often hear this term called apoptosis. Now this apoptosis is actually cell death. Okay, programmed cell death. That means it's a cellular suicide mechanism, highly regulated cell death mechanism. But before we go deeply into apoptosis, we will study about two types of cell death. So there are actually two types of cell death. One is called necrosis or accidental cell death, and the second one is, yeah, programmed cell death or apoptosis. Now, when we say about necrosis, accidental cell death, we can say that this type of cell death happens when cell is subjected to any injury or when the cell is in excessive amount of stress. At that time, yeah, cellular membrane they swell and they become permeable, and the lytic enzymes destroy the cellular content and all the cellular, and you can see all the contents leak into intracellular space. That type of death is actually necrosis, also known as accidental cell death. The one that we are going to discuss in detail is apoptosis. It is also known as PCD, program cell death. And yeah, it is unique to animal cells. That means there will be no program cell death in plants. It is unique to animal cells and it's a highly regulated mechanism. And yeah, it is cellular suicide. It is activated from, yeah, activation of so many intercellular program. Okay, and it doesn't happen often. Whenever there is any problem in the cell, just like DNA damage or abnormal cell division or disruption of cytoskeleton, virus infection, on those time only cell actually activates this mechanism called program cell death mechanism, that is called apoptosis. So here I would like to show you what are the activities happening in the apoptosis. Okay, so this is the cell which is shown in the slide, which will undergo apoptosis. So what happens is, maybe due this cell was having any problem, so a death signal came to this cell, and when the signal came, apoptosis mechanism becomes active, and you can see the cell shrinks in size, and yeah, it's chromatin condensed, and cell actually breaks into so many small membrane bound bodies, and those small membrane bound of bodies are called apoptotic bodies. And yeah, these apoptotic bodies are then engulfed by nearby cell, leaving no trace in the intercellular spaces. Okay, while as in necrosis, yeah, you can see the cellular content leaking in the intercellular spaces. But in apoptosis, it's a programmed cell death. So cell, when it shrinks in size, fragments into small membrane bound bodies, it is very nicely engulfed by nearby cells, leaving no trace in the intercellular spaces. Okay. Now, what are examples of some cells which undergoes apoptosis? So, if there is excess cell in body and those excess cell, if they are having no function in the cell, yeah, those type of cells will be, yeah, undergoing apoptosis, because cell body will not like to keep those cells which are having no functions. Then virus infected cells are another example of cells which can undergo apoptosis. Then those cells which are not producing ATP properly, those cells will be also undergo apoptosis. Then those cells which are having uh, disturbed cell cycle, that type of cells will be also undergoing apoptosis. So that is about apoptosis. It's unique to animal cells. It's a cellular suicide mechanism. And yeah, It is highly regulated. Okay. Now we will study about three major factors contributing in our apoptosis program. So there are three major proteins present in apoptosis. First one is an enzyme called caspase enzyme. Okay. Caspase enzyme. So caspase is a short form. These are protease enzyme. That means they can target and cleave so many different protein substrates. So more than 100 types of protein substrates are actually cleaved by caspase enzymes. So caspase, cysteine aspartases specific proteases. That is a full form of caspase enzyme. These are caspase are actually protease enzyme, and they are having a cysteine amino acid in their active site. And the target substrate of caspases, yeah, when they are identified by caspases, they cleave on their specific aspartic acid site, and that's why the name came cysteine aspartases specific protease. So these are protease enzyme with, yeah, a key cysteine residue in their active site, and they cleave the aspartic acid site of target substrate proteins. That's why the name. Now there are uh, two different types of caspases enzymes. Initiator caspases, which are activated first during apoptosis pathway, and yeah, examples of initiator caspases are caspase 2, caspase 8, caspase 9, and caspase 10. Then the second caspases are effector caspases, also known as executioner caspases. So these effector or executioner caspases are the one which cleaves more than 100 different types of protein substrate. And the examples are caspase 3, caspase 7, and caspase 6. Okay. So initiator caspases and effector caspases. Then all the caspase enzyme, whether initiator or effector, all the caspase enzyme have a pro domain, okay, in their structure, or these enzymes will have a pro domain which makes them inactive. So all the caspases are inactive in the cell until their pro domain is cleaved. They will never become active, and if they become active, then only they can do their function. So all the caspases will have pro domain, and this pro domain is cleaved first to become an active caspase enzyme. So these are the first major factor in apoptosis. Caspases, proteases enzyme, they are protease enzyme with key cysteine residue in their active site, and they cleave their target protein at their specific aspartic acid site. That's why the name. And yeah, there are two major classes: initiator caspase, which becomes active first, and initiator caspase will activate effector or executioner caspase, and executioner or effector caspase will then cleave the substrates. Okay, so in this picture, it is shown that, yeah, the pro domain, this pro domain is cleaved to become mature caspase. Yeah, some of the functions of caspases are mentioned. Yeah, it fragments the DNA, it also fragments the nucleus, disrupts the cytoskeleton, and yeah, it also fragments Golgi bodies. So in short, we can say caspase can actually uh, target uh, almost all the cellular substrate. Then the second major factor in apoptosis is the mitochondria, the cell organelle mitochondria, which gives the cell life by producing ATP. But the same mitochondria helps in, yeah, program cell death mechanism also. So if we see the structure of mitochondria, we know that it is a double layer membrane. It will have an outer membrane, and it will also have an inner membrane. And in between outer and an inner membrane, there is a intermembrane space. So in this intermembrane space of mitochondria, so many molecules are present, like cytochrome C, SMAC, AIF, apoptosis inducing factor, and endonuclease G. So all these molecules are activators. They activate initiator caspase. Okay, and we know that initiator caspase will finally activate effector caspase, and effector caspase will cleave the cell substrate. So yeah, so all the molecules which are dangerous to cell or which promotes apoptosis are yeah, actually inside this intermembrane space of mitochondria. So mitochondria which produces ATP, yeah, the same mitochondria also guards those molecule which activates caspase enzyme. Okay, initiator caspase enzyme, and they are present in intermembrane space of mitochondria. Okay, so uh, once this molecule escape from intermembrane space of mitochondria to cytosol, yeah, cell will die or program cell death or apoptosis can become active and the cell will die. Then the third major factor involved in apoptosis are BCL2 family protein. So the BCL2 stands for uh, B cell lymphoma 2 family proteins. BCL2 stands for B cell lymphoma 2. So now this BCL2 family proteins, they are also present in yeah, all the mammals and vertebrates, and yeah, they also help in apoptosis. And generally, in us mammals, there are approximately 20 related BCL2 family proteins, and all these 20 BCL2 family proteins, yeah, are divided into three functional groups. Okay. Before that, I would like to mention that BCL2 family proteins, yeah, they are all three functional groups are having BH3 domain. Okay, BH3 domain. So this BH stands for BCL2 homology domain. So BCL2 family proteins will have BH domain. Either one, at least one they will have. Okay, at least one or three or four. But yeah, all the BCL2 family proteins are having this BH3 domain. Okay, and you can see BH3 domain is common in all three. Okay, and we will discuss about it later. So BCL2 family protein, third major factor involved in apoptosis. Yeah, so they are further divided into three functional groups or functional classes. First one is anti-apoptotic proteins, also known as the protectors. Okay, and from the name itself, we can say those proteins which actually inhibits or protects the cell from apoptosis are anti-apoptotic proteins or the protectors. So yeah, so these are some of the examples. Two most common examples are BCL2 and BCLXL. Okay. Then the second uh, functional group of BCL2 family proteins are pro-apoptotic proteins or killers. Yeah, and again, the name suggests that they promote apoptosis or they promote yeah, this program cell death. And two most common examples are BAX and BAC. Okay. Then the third ones are called BH3 only proteins or BCL2 regulator proteins. And that's why the name regulator. And they also promote apoptosis by either inhibiting anti-apoptotic protein or directly activating pro-apoptotic protein. Okay. So some of them, some common examples are BAD, BID, BIM, Puma, Noxa. And yeah, among the three, anti-apoptotic protein and pro-apoptotic proteins are found on outer mitochondrial membrane. And the third one, BH3 only protein, is found in cytoplasm. Okay, remember I note it down. So yeah, all of them are having BH domain. At least one BH domain they will possess. Okay, then how anti-apoptotic protein helps in protecting the cell from apoptosis. So you can see that they all have one domain common, that is the BH3 domain. So yeah, they can bind to each other. And yeah, we know that pro-apoptotic proteins are killers. They promote apoptosis. And pro-apoptotic protein, when they are free, so many free pro-apoptotic monomer proteins can uh, combine together, oligomerize, and forms a channel. And remember, it is on outer mitochondrial membrane. So these are present on outer mitochondrial membrane. So if they oligomerizes, they form a channel like opening, and from that channel like opening, yeah, these cytochrome C and these things can escape from intermembrane space of mitochondria to cytosol. And once they are escaping from mitochondria, they will leak in the cytosol, and in the cytosol they will activate caspases, which will lead to cell death. And that's how pro-apoptotic protein works. They so many monomer pro-apoptotic proteins, they oligomerize and forms a channel like opening, and through those channel, all those proteins which activate caspases, they come out from intermembrane space of mitochondria. So how does anti-apoptotic protein inhibit apoptosis? So this anti-apoptotic protein, which is having BH3 domain, they go and they bind to pro-apoptotic protein on their BH3 domain, not letting them oligomerize, and that's how they uh, promote or protect the cell from apoptosis. Okay, so anti-apoptotic protein binds with pro-apoptotic protein so that they do not oligomerize. Okay, now we are having BH3 only proteins. Now these BH3 only proteins, they promote apoptosis. So they can directly go and activate pro-apoptotic protein. Okay, they can bind and they can make them active. Or what they do is they will go and they will bind to anti-apoptotic protein by their BH3 domain. And when BH3 only protein bind with anti-apoptotic protein, it is unable to bind with pro-apoptotic protein. So many pro-apoptotic protein will oligomerize on outer mitochondrial membrane, and from there it will form a channel-like opening. And from that channel-like opening, all the proteins which activate caspases will come out from intermembrane space of mitochondria. So that's how they work. Okay. Okay. Then, okay. Suppose, yeah, you can see normal cell. This is a pro-apoptotic protein, and yeah, anti-apoptotic protein is binding them. So no cell death take place. So here it is written apoptotic cell death signal came. Yeah. So pro-apoptotic protein, yeah, BH3 only protein will come and bind with anti-apoptotic protein. So this, yeah, here you can see. So pro-apoptotic protein is free, and yeah, it will promote apoptosis. So basically, whenever cell death signal comes, BH3 only protein will go and bind with anti-apoptotic protein, letting all the pro-apoptotic proteins free, and they will form channel-like opening in the mitochondrial membrane, outer mitochondrial membrane, and through this outer mitochondrial membrane, so many proteins like cytochrome C, it will come out and it will bind with a adapter protein called APAF1. Now this APAF1, it looks like a wheel-like structure. So cytochrome C, when they come out from intermembrane space, will bind to this APAF1, and this structure is known as apoptosome or death wheel. Since it looks like a wheel-like structure, it is also known as death wheel. Now, our cytochrome C and APAF1 has formed apoptosome. So this apoptosome activate initiator caspase. It cleaves the pro-domain of initiator caspase and it becomes active initiator caspase. Okay, remember I told you all the caspases will have this pro-domain, and this pro-domain has to be removed. Then only caspases will be activated. So once cytochrome C bind with uh, our APAF1, it forms apoptosome. Apoptosome will cleave the pro-domain of initiator caspases. So this, yeah, caspase 3, we know that caspase, initiator caspases like 2, 8, 9, and 10. So here you can see it is written caspase 9. So whenever cytochrome C combines or binds to apoptosome, yeah, here caspase 9 is shown, it's a initiator caspase. So it will activate, it will cleave its pro-domain, and caspase 9 will become active. And caspase 9 will go and cleave the pro-domain of effector caspase. This one here shown is caspase 3, and we know that caspase 3 is an example of executioner or effector caspase. So active initiator caspase will cleave the pro-domain of effector caspase, and yeah, effector caspase will go and actually degrade all the cellular substrate, finally leading to apoptosis or programmed cell death. So this is how generally apoptosis works. Whenever a death signal comes due to any circumstances like DNA damage, virus infection, or absence of growth, or due to uh, bad cell cycle mechanism, yeah, cell cell death signal will come, and that cell death signal will let the BH3 only protein go and bind with anti-apoptotic protein. So anti-apoptotic protein binds is then uh, not able to bind with pro-apoptotic protein. So they oligomerize on mitochondrial membrane and forms a channel-like opening. And through this channel-like opening escapes our cytochrome C. And cytochrome C, once they are inside the cytosol, they combine with or they bind with APAF1, forming a death wheel, and it is known as apoptosome. Apoptosome will activate initiator caspase. Initiator caspase will finally activate effector caspase. Effector caspase will degrade all the cellular substrate, leading to cell death. So this is about introduction to apoptosis, factors involved in apoptosis, and yeah, general mechanism of apoptosis. Thank you.